Thank you so much. Thanks for having me, and thanks for inviting me to talk about something that's dear to my heart, and it's an old new friend, and that's porcine factor VIII. It's been around for a while, and I'm going to talk about some of the history and where we're going. These are my disclosures. Of particular interest is that I have been very involved in the phase 2-3 clinical study that led to the approval of recombinant porcine factor that is now owned by Baxalta. So where does this whole idea come from? And this is an older slide, but it shows in red people with anti-human inhibitors, and they're listed in increasing order here. And in black, you see the correlating cross-reactivity to an anti-porcine inhibitor. And the observation was that just because you have a human anti-factor VIII inhibitor, you don't necessarily have a porcine inhibitor. So therefore, would that be a good treatment in inhibitor patients? So this led to the first commercial production of a plasma-derived porcine factor known as High-8C. Initially, porcine factor was being produced, or porcine plasma was used because it was more abundant, and then it was lyophilized, highly purified, and then was licensed first in the UK and then in the US in 1986. This was not a very pure product. It was plasma-derived. 1% of this product was actually factor VIII, and the rest were other proteins, such as von Willebrand factor. It was used very nicely and very effective in patients with inhibitors, both acquired and congenital hemophilia. But maybe because of the high von Willebrand factor content or some other reasons, there was a dose-dependent thrombocytopenia that was noted with the use of High-8C. There were also no viral inactivation steps, and in the late 90s, low levels of parvovirus were found in the product, and in 2004, manufacture of High-8C ceased. During the same time, the thought of a recombinant porcine factor came about, and that's the protein, or this is the product I'm going to be spending the rest or the remainder of the talk about. In structure, it has the same domain sequence as human factor VIII. It binds very well to human factor, von Willebrand factor, even though recombinant, the factor concentrate does not contain von Willebrand factor. It's a B-domain deleted product. And there is some divergence in the amino acid sequence. There is about an 86% homology, but especially here in the A2 and the C1 region, C2 region, which we know are quite immunogenic, there is not as much homology, and maybe that's the reason why you don't get this cross-reactivity between human and porcine factor. It is manufactured in a baby hamster kidney-derived cell and grown in cell culture medium. It undergoes 2 purification steps, and therefore there are no extraneous mammalian proteins, and from an infectious risk, it's thought to be a very safe product. Does it work? This is some work from Claude Negrier, who looked at this recombinant porcine factor and gave it to a patient right here that had a high human factor VIII inhibitor and a low recombinant porcine inhibitor, and saw that in a dose-dependent fashion you get increasing thrombin generation. And if at the same time you look under electron microscopy, you also see a dose-dependent optimal clot quality obtained in these samples. So it seems to be working, at least in vitro. Can you measure a factor VIII with that? This is a study now going closer in time that Baxalta did. That was a collaborative assay field study to see whether we can get reliable factor VIII levels when we administer recombinant porcine factor. Recombinant porcine factor is now licensed under the name Obizur, and this was compared to a recombinant factor VIII, in this case Advate. So this was a blinded study. Samples were spiked with different amounts of either Obizur or Advate and were sent to several laboratories around the world. And these are the results. This was a sample spike to give you about an 80%, 20%, and 5% assay. And here you can see that Obizur really performed pretty much as well as Advate would. So it gives us pretty reliable factor levels in that range. Little less so in the 20% range, but still very good correlation. And then as we get into lower ranges, just as we saw with the Advate, maybe it's not quite as reliable, but we're still getting factor VIII levels. How does that all influence recovery of this product? And I'm now going to draw from different experiences, and some of them are from clinical trials or early clinical trials in congenital hemophilia, and then I'm also drawing from the data in acquired hemophilia. They're not the same disease. So I'm going to point out where I'm talking about which one. But this is a product that is now licensed in the U.S. and Canada and several European countries for acquired hemophilia, not for congenital hemophilia. But I'm quite sure that we're going to start seeing its use in congenital hemophilia as well. So the recovery we can expect, or that was seen in congenital hemophilia in an early phase 1 PK study done by Christine Campton et al., showed that 1 unit of this recombinant porcine factor gave you a main recovery of 1.76 amine, but with a really wide range, as you can see. And that's very important to know about this product. You just don't know what your recovery is going to be. So what about acquired hemophilia? And this is data drawn from the phase 2-3 study in acquired hemophilia. Here again, 1 unit of the recombinant porcine factor raised the factor VIII level by a mean of 1%. But again, we saw a very, very wide range, where one patient only went to 10% and one went to 261. That's a huge difference. Well, that makes sense because we're talking about inhibitors. So if we're taking the inhibitors, the anti-porcine inhibitors, cross-reacting inhibitors out, we still see a very wide range. And that's something we need to be aware of as we're using this product. How about half-life? I just pulled some of the patients from the acquired hemophilia trial, some of them that did not have a recombinant porcine factor inhibitor, like the 2 patients up here, and some that did have cross-reactivity with a recombinant porcine inhibitor. So here you have a patient that responds really nicely, goes up to about 200, and then has a half-life of almost 12 hours. That's beautiful. What about this patient? Well, this patient also goes up very nicely, but only has a half-life of about 5 hours. So both of them did not have underlying porcine inhibitors, but yet I get this huge range of different responses. And if I have the patient in front of me and I need to treat them, I don't know what I'm going to have. And as you're going to see, there are no predictive factors. What about these patients that do have underlying porcine inhibitors? Well, they have very, very short half-lives of about 2 to 3 hours. So that's something, yes, you can get nice factor VIII levels if you give enough recombinant porcine factor, but you have to dose it much more frequently. What about the clinical efficacy of recombinant porcine factor? We have a phase 1 study. I talked about that. I'm going to talk a little bit about the phase 2 study that, to my knowledge, really has not fully been published yet. So I'm just going to draw from what has been presented at previous meetings in abstract form. Then I'm going to talk, this is in congenital hemophilia, I'm going to talk some about acquired hemophilia and the study I participated in. And clearly, these are studies of very, very few patients, and we're really looking forward to the post-marketing studies to show us more about how to use this product. So let's go to the congenital hemophilia first. Again, this comes from a small trial. These were 9 patients with congenital hemophilia with inhibitor, 8 of them severe, 1 moderate, and they were treated for non-life or limb-threatening bleeding. They were aged 14 to 34, and 100% of these bleeds were deemed to be treated successfully with good efficacy. And down here, in red, I teased out what these patients' underlying human inhibitors were, and they were not very high. One was up to 15. And in green, you see the patient's anti-recombinant porcine inhibitors. So some of these patients did have some porcine inhibitors. Oops. Let me go back one. So these were the bleeds. Some of these patients, like this patient here, had 8 bleeds treated, all of them successfully, and all of them with only 1 dose of the recombinant porcine factor. Some patients here, and this is one who has a higher recombinant porcine factor, needed up to 8 doses. But again, all of them were treated in the end successfully. Let's go to the acquired hemophilia study, the larger study that enrolled 29 patients. These were adult patients. As acquired hemophilia, you would expect. We saw an older age range. These patients were hospitalized this time for severe bleeding, severe life or limb-threatening bleeding, and they got a bit of a higher dose based on the fact that the range, as I showed you, of recoveries are very, very variable, and these are life-threatening bleeds. Then they were observed with factor VIII levels and clinical assessment every 2 to 3 hours, and then at 24 hours was the primary endpoint to see whether we achieved hemostasis. 100% of these bleeds were controlled at 24 hours, as you're going to see. And here it is. One of the patients of the 29 patients turned out not to have hemophilia, so he was excluded from the analysis. But of the 28 patients that did actually have acquired hemophilia, all of them had good hemostasis, 24 with complete cessation of bleeding, and 4 with significant reduction in bleeding at 24 hours. What about those factor VIII levels? I showed you that the recovery can vary drastically. So let's first look at the response to the very first dose of 200 units per kilogram. The response was anywhere from 20 to 540% factor VIII activity level. That scared some of us tremendously. How did that go on over the max? Some of them were actually, by repeated dosing, got factor VIII levels as high as 775. But everybody, even patients with inhibitors, as you're going to see, achieved levels over 100%. That was all controlled with subsequent testing, and patients were well controlled at 24 hours with an acceptable range. So how were these low or very high recoveries? What could predict what's doing that? And obviously, it depends on what your anti-porcine inhibitor is underlying that. If your anti-porcine inhibitor is 0, a dose of 200 units per kilogram is definitely going to get you into a very safe range. It may be a range that is too high for your comfort, and maybe this is where you want to start with a little bit of a lower dose. Let's now look at people that had cross-reactivity, and clearly these are the people that are not going to respond as well. So it's very important to know when you are treating your patient with acquired hemophilia whether you are dealing with cross-reactivity or not. I also looked at whether the actual anti-human inhibitor had any predictive value in how people would respond, because there is some notion from the VIII experience that maybe the higher anti-human inhibitor would also have cross-reactivity, and therefore people would have lower recoveries. That was not true, and here you can see that people with a little bit higher human Bethesda units actually had a higher recovery than people with lower. Very, very small numbers, but not a predictor. What about safety? As in anybody with acquired hemophilia, there's an older population with lots of comorbidities. We saw a lot of adverse events. Only 2 of them were related to the study or to the product, and both of them were a development of an anti-porcine inhibitor. There were 33 serious adverse events, 7 deaths, but none of them were deemed to be due to the product or the study. So as we're talking about these inhibitors that develop on study, let's take a look at first the acquired hemophilia and then a little bit the congenital hemophilia data. These are all the 28 patients that entered the acquired hemophilia trial, and at baseline, 18 of them had no cross-reactivity with a recombinant porcine inhibitor, but 10 of them did. These 10 of them, we did not know the numbers at the time we started treating them. These were people that had serious life-threatening bleeding, so we just started the treatment. And remember, all of them responded very nicely, even though we had somebody there who had a porcine inhibitor of 29 Bethesda units. Of these 18 patients, 5 of them eventually over 90 days developed an inhibitor. Of these patients, 8 of them actually started losing their inhibitor on treatment, and 2 of them persisted. Oops, there's a slide missing. Oh, here we go. This is the very, very limited data of the congenital hemophilia, and I wanted to bring that up because it's important for us to know what is the inhibitor risk in people with congenital hemophilia, and is it different? So here going into the trial, these are the 9 patients. 4 of them did not have cross-reactivity. 1 of them was kind of equivocal, and 4 of them did have underlying porcine inhibitors, and this is how high they were. Those 4 all persisted. None of them went away during treatment. Of these 5 patients, 4 of them eventually made anti-porcine inhibitors. So this is a product that is probably very efficacious, and it's a great product, but eventually, especially in congenital hemophilia, I assume we're going to have to think that these people are going to get anti-porcine inhibitors, and that we then no longer can use this product. Very responsive here. So I just wanted to finish on a couple of slides that are a practical consideration with this product. I think, and I'm not getting paid for this, but I think it is an efficacious product. You can get measurable factor VIII levels, and it is great if you have somebody who has a life-threatening bleed. It's certainly something that I would pull off the shelf as a first-line treatment. But let's talk about the cost a little bit. This is a 76-year-old gentleman that presents with right hand swelling. His finger and thumb are numb, and they're cold to touch. So I'm really worried about compartment syndrome. This is his right hand. He's right-handed. Letting this go can really result in a lot of comorbidity for him. So what am I going to treat him with? I decided to give him recombinant porcine factor. I decided to give him a little bit of a lower dose than what was usually in the package insert. And he went up very nicely to a comfortable level, or even a little bit high, and then I just kind of let it drift down. And he had a very nice half-life, didn't really have to treat him for the next 24 hours again, so that one dose did really well. His swelling went down, his feeling came back, he started feeling better. I gave him one more healing dose, really just in case, before I sent him home, and he recovered very nicely. What does that mean in cost? And these are the costs for the products that are licensed in the United States. The costs are going to be different, but in different countries, but they're somewhat similar. And I'm just bringing this up so you start thinking about that as you're using that in your countries. So Obizur is the recombinant porcine factor that's licensed in the United States, and in the first 24 hours I would have spent $41,000. If I would have treated him with VIII, which is a very good alternative, these would have been the prices for an equivalent dosing. I probably would have done VIII every 2 hours for about 3 doses and then dialed it back, so probably you would have used 9 doses over the first 24 hours. And depending on what pricing you have, you can see that would have been a lot more expensive. What about fiber? Probably would have used 75 units per kilogram for 4 doses, giving it every 6 hours, which I think is an appropriate treatment. And here you can see, depending on what pricing, and these are the hemophilia treatment prices, these are the normal prices, but I really, with the Obizur at this point, I may come in under cost with a great product, with great efficacy, with something with factor VIII level I can measure. Sounds great. What about that person that I showed you in the clinical trial that had a anti-recombinant porcine factor of 29 Bethesda units? Well, I need a lot more product here. So I'm giving 200, and a couple hours later, 300, and 400, and 400, and 400, and 400 units per kilogram. So let's do the cost analysis there. This patient would have cost $766,000 in the first 24 hours. So yes, this patient was treated for a life-threatening bleed and achieved good hemostasis in 24 hours, but at a cost, if I'm comparing that to what else I could have treated this person with, that I cannot stomach, that our healthcare system cannot stomach. So that is something where the product may become cost prohibitive, and we have to do something about that. So what we've done in our hospital, what other hospitals have done, is they have calculated what would be the equivalent dose that to either Novo-7 or to FIBA, to the alternatives that make it cost-effective to use this product. And we came up that it's probably going to be 5 doses of 100 units per kilogram in the first 24 hours. But every hospital kind of is going to have to calculate that by themselves. You get into deep water if you just start using it and you don't start thinking about the costs. Now I want to applaud Baxalta, who recently or last week came to me and said that they're thinking about a cost strategy to bring down the cost of this product, and hopefully we're going to be able to use more of it in the future. So in summary, this is a recombinant porcine factor. It is very hemostatic. You've seen that in the clinical trials. In both the acquired hemophilia as well as congenital hemophilia. You can monitor factor VIII levels quite reliably. We have not seen any allergic reactions. We have not seen any thrombocytopenia like we did with the high 8C. Some cross-reactivity exists at baseline, but still we could treat these patients. But it definitely will affect the recovery and half-life. So you have to monitor these patients frequently. Most of the patients did develop anti-recombinant porcine factors after exposure eventually. So it's an effective hemostatic agent in patients with inhibitors. Its effects are very dynamic. It's potentially cost prohibitive, but hopefully in the future the pricing scheme is going to be better and we're going to learn more about it and how to use it. Thank you.